I'm Dr. Ray Chan, one of the scientists at Philips Research North America. I've been working with multimodality cardiovascular imaging for over 15 years, both in academia and industry. Time and again in our research projects on the development of either new image acquisition techniques or novel processing methods, we've run into the need for versatile imaging targets that can be used reliably and reproducibly to mimic biological organ behavior under ultrasonic, optical, X-ray, and magnetic resonance-based interrogation. In this video segment, you'll see a novel multimodality phantom that we've developed for our experiments focused on the imaging of cardiac structure and function. You'll see an overview of its key properties and a range of different applications in which it's being used for research activities at the lab. For the particular needs of our research projects, the first version of the cardiac phantom that we developed has a geometry that mimics the anatomy of the left and right ventricles, as well as an abstract representation of coronary vasculature. The tissue substrate used for the organ body is a hydrogel with tuned stiffness characteristics that approximate the biomechanical properties of soft tissue. The hydrogel itself has also been doped with contrast materials to allow it to appear like soft tissue under ultrasound, X-ray, CT, and MR imaging. Cardiac motion with the organ phantom is imparted by computer-controlled actuators that allow for independent control of compression, stretching, and torsional modes of tissue deformation. Standalone software with application program interfaces for further customization can be run on a laptop to control the actuators that we use. Actual ECG signals can also be used as inputs to drive the timing of contraction, and this scheme is flexible enough to allow for regular RR intervals to be simulated. An MR-compatible version of this actuator assembly based on computer-controlled pneumatics has been developed and is currently undergoing further testing. In the current model, we use silicon-based tubing for the overlying vessels, and the entire organ phantom is mounted within an acrylic housing. That can be used as a water bath during acoustic testing or for interventional experiments which require device manipulation within a water medium. The acrylic body of the phantom housing is versatile and reconfigurable. It has standardized ports for fluid flow, drainage, and interventional device access. Various acoustic windows are also available for multi-angle ultrasound imaging, as are mounts for additional imaging targets that can be used in characterization of resolution, contrast sensitivity, or localization accuracy of interventional devices. Our ultrasound-based experiments on the cardiac phantom focus on characterization of the ability of different imaging modes to assess structural anatomy, dynamic biomechanical behavior, and hemodynamics. I will demonstrate imaging of phantom under M mode and tissue Doppler imaging, then B mode imaging, and finally 3D ultrasonography. Here in M mode, you can see the A line of interest selected from a two-dimensional ultrasound image, and below you can see the A line behavior as a function of time. Enabling tissue Doppler, you can see the tissue velocity displayed as a color overlay, and from these imaging modes you can appreciate the displacement of the phantom structure at opposing walls as a function of time. Switching to B mode imaging, we can see the structure and dynamics of the phantom within any 2D slice of interest, and also, turning on color Doppler, we can visualize fluid motion within the phantom chamber. Finally, with real-time 3D imaging, we can visualize and quantify volumetric structure and dynamic behavior, and in addition, we can manipulate interventional devices being moved within the chamber anatomy under image guidance. In all of these imaging modes, it is clear that the appearance of the phantom material resembles the intensity and speckle pattern expected of biological tissue, and that the range of dynamic behavior also approximates that of the beating heart. We have used these capabilities to validate new algorithms for segmentation, for registration of multiple volumetric acquisitions, and for tracking of tissue motion within the phantom walls. The reproducibility and multimodal nature of our phantom allows us to compare ultrasound-derived results with reference measurements from sonometry, cardiac CT, and magnetic resonance imaging.
Hi, my name is Dr. Luis Gutierrez, and I've been working in multimodality image guidance for 4 years. During this time, I've focused on catheter-based interventions, which are primarily guided by X-ray fluoroscopy imaging. As is seen here, without any contrast agent, the heart shadow or phantom is only faintly visible on X-ray imaging, just like in real cases. The ports in the vessels and chambers allow us to insert catheters and visualize their movement under fluoroscopic imaging. These catheters can also be used to inject a contrast agent, as shown. The contrast agent allows us to selectively visualize the vascular space that has been filled, while leaving the rest of the heart with its normal image appearance. This ability to selectively opacify the coronary vessels allows us to test new X-ray technologies like rotational imaging. Rotational X-ray fluoroscopy is a new technology that allows for a number of new ways to see structures in the heart. One of those is 3D vessel modeling, in which the contrast images of the coronary vessels are used to generate a three-dimensional model that the physician can look at from any orientation without having to continue exposing the patient to more X-rays and contrast agent. Finally, because the dynamic heart phantom has multimodality contrast properties, we can use it to evaluate new ways of combining X-ray fluoroscopy with different imaging modalities, such as ultrasound as seen here. The development of these new technologies like 3D vessel modeling and multimodality imaging require a significant amount of testing and retesting in the lab, which is made much easier by using a realistic dynamic heart phantom. Without such a phantom, we could try to acquire real data in hospital settings, but this requires getting IRB-approved protocols, which usually means lots of red tape, and then waiting for suitable patients to volunteer. Having our realistic phantom handy in the lab saves us lots of time and money in the development process.
Hello, my name is Dr. Robert Manske. I have been working with Philips research on cardiac CT reconstruction algorithms for multi-slice cone-beam CT scanners for 6 years, introducing some of the most powerful reconstruction algorithms available. I would like to discuss an application of our dynamic motion phantom in which it was used to simulate cardiac motion artifacts in CT. The phantom's ability to reproduce realistic cardiac motion helps us to develop and test novel reconstruction and acquisition strategies in order to get the best possible image quality. By activating the phantom's computer-controlled actuators during the acquisition of CT image data, we can produce motion artifacts in the reconstruction. As you can see here, the reconstructions suffer from realistic cardiac motion artifacts, which manifest, for example, as blurry cardiac chambers and vessel shadowing. It is critically important in cardiac CT applications to be able to accurately delineate the coronary vessels. In this image of the cardiac phantom, which is suffering from motion artifacts, the vessels modeled on the cardiac surface cannot be delineated clearly. This is an ongoing challenge in cardiac CT imaging. To be clinically valuable, coronary arteries with a diameter below a millimeter need to be reconstructed during coronary CT angiographies. With our phantom, we can model cardiac motion and vessel displacement, generating artificial CT data and work on improvements of our reconstruction algorithms.
I'm Dr. Daniel Elgort. I've been developing MRI imaging and reconstruction methods for diagnostic and interventional cardiac applications for nearly 10 years. The primary challenge for these MR researchers continues to be that many of the important cardiac anatomical structures are small, geometrically complex, and of course, they are constantly moving. This phantom provides a realistic model of the human heart that is capable of reproducing these critical features. This heart phantom accurately depicts the complex geometry of the human heart at a 1:1 scale. Doping the phantom's construction material with an MR contrast agent allows its relaxation and contrast properties to mimic the properties of actual human myocardium. Using MR-compatible pneumatic actuators, the phantom is able to exhibit realistic motion inside the scanner during data acquisition. The software which is used for motion control even enables the simulation of irregular heart rhythms with a non-constant R-to-R interval, which is the source of poor image quality for many existing motion compensation techniques.
You've seen a version of our multimodality dynamic heart phantom whose design specifications were chosen to meet the immediate needs of our research projects. These requirements were accurate representation of cardiac ventricular anatomy, a coarse representation of coronary vasculature and compression, as well as torsional modes of dynamic behavior. You've seen a demonstration of its use for ultrasound, X-ray, CT, and MRI applications. The capabilities provided by this phantom have reduced our reliance on animal studies and human volunteers, and is a very cost-effective tool for shortening the research and development cycle when designing new methods for image-based diagnostics and interventions. This multimodality dynamic heart phantom can help to increase the pace and productivity of your research projects in medical imaging.